right, time for our last topic, excretion in insects. I suspect you might be thinking, well, what's the big deal with excretion in insects? In other words, why should I care? Here are some thoughts about that kind of tie into thoughts about studying comparative physiology in general. When we each find ourselves in situations where we have to problem solve, such as when the cat walks across the screen, one of the resources we rely on is things we know or have learned. So if we had focused all of our energies on studying the mammalian kidney function in all of its granular detail, well, that's great, but it misses the point that other animals have equally interesting, creative, and different strategies for solving the same kinds of environmental challenges. So in the long run, I think we benefit from studying comparative physiology because that gives us a more versatile collection of strategies to consider, and therefore more perspective on how any single strategy may have evolved or come about. On that note, throughout the semester, we've seen that terrestrial insects have a range of characteristics that make them physiologically quite distinct from other animals. The biggest component has been the decoupling of gas exchange from circulation, which we've already discussed quite a bit. But now if we're talking about osmoregulation, this is going to come into play again. And why is that? Well, if we think back to how mammalian and even amphibian kidneys work, and as a side note, bird kidneys are kind of like hybrids between amphibian and mammalian kidneys, with just a few twists at the end. Anyway, so if we think back to how mammalian and amphibian kidneys work, let's think about what the first step is in urine production. It's ultrafiltration due to the pressure differential between the glomerulus and Bowman's capsule, right? So now coming back to insects, now we've got an animal with an open circulatory system, right? So therefore, this is a low pressure circulatory system, and therefore we can't drive urine production via ultrafiltration. So instead, a mechanism as mentioned back in video 10.9 comes into play, and that is filtration via active solute secretion. So let's take a look at some insect anatomy in closer detail. So here is an artist's rendition of an insect's posterior end, well, the internal parts of the posterior end. So up top here is the insect's midgut, and the Malpighian tubules shown here are those organs that are responsible for primary urine formation. I'd note that the number of Malpighian tubules varies across insect species from anywhere between 2 to up around 200. And just for some reference, the crickets that we worked with in lab have around 5 pairs of Malpighian tubules. Okay, so if these guys use active solute secretion to get fluid and other solutes to move into the Malpighian tubules, do you have any guess as to what solute gets actively transported in? Well, if you were to guess sodium and chloride, you would be wrong. Well, okay, half wrong, most of the time. There are always little exceptions. Instead, in general, insects secrete potassium chloride. And they do this at a rate that's so high that the potassium concentration in the tubular fluid can be 6 to 30 times higher than in the blood. So this mechanism is one that works via secondary active transport. And it involves a proton pump, but we don't need to be overly concerned with those details at this point. Instead, just know that the flux of potassium chloride into the tubule lumen drives the osmotic entry of water, and that happens pretty quickly, such that the fluid remains roughly isosmotic to the hemolymph. Other solutes then follow, so that includes amino acids, sugars, organic wastes, and toxins, and several inorganic ions also. And as with the blood in other animals, larger proteins are generally excluded from the Malpighian tubule urine. However, many insects can also actively secrete other sorts of organic compounds, such as detoxification products or plant defense chemicals as well. There can also be reabsorption as fluid moves down the Malpighian tubule towards the gut. But overall, in the end, the fluid that reaches the hindgut is more or less isosmotic, to the hemolymph, and it's going to contain solutes that differ in composition from the hemolymph. Specifically, the fluid's going to be very rich in potassium chloride due to this secretion mechanism. So now, as with mammals, insects generally produce a substantial volume of this primary urine. But they don't excrete this much urine overall. So instead, the next events take place in the hindgut, and particularly the rectum, where urine composition, concentration, and volume all get modified. So for example, the rectum can reabsorb a whole host of substances. So it can reabsorb water, potassium, sodium, and chloride, and also amino acids, acetate, and phosphates. And in the meantime, rectal cells can also secrete hydrogen ions. And this acidification will cause uric acid and urates to precipitate out. And not only can the rectum do these things, it can also regulate the relative rates of reabsorption of water and solutes and can adjust the urine ion composition, right? So it's not just reabsorbing, but it's also regulating. And even further, insects use a unique mechanism termed local osmosis to continue removing water seemingly against a large and opposing osmotic gradient between the rectal fluid on the inside and the hemolymph on the outside. And this happens even in the absence of simultaneous solute reabsorption. So this is distinct from other processes we've seen. I'm going to leave the details of how local osmosis works as a mystery for the moment, but there's a lot more on this topic in chapter 29 of your textbook if your curiosity has been piqued. For now, it will just suffice to say that this is accomplished by cells in these rectal pads here. And things can get even weirder in that some insects can even use specialized cells in the rectum to produce extremely concentrated excrement pellets to the extent that they are effectively dry. And often that's referred to as frass. The cell complexes that accomplish this can also allow some insects to absorb water vapor directly from the air, right, through their butts. And at this point, you will probably never think about insect rectums in the same way ever again. Anyway, much as I might like to go on about this, we need to move on so we can cover our final topic, nitrogen disposition and excretion. So why this topic? Well, if you think about it, we've spent plenty of time already this semester thinking about other metabolic end products, primarily carbon dioxide and water. But the fourth most abundant atom found in animals is nitrogen. And nitrogen, is a fundamental part of proteins and nucleic acids. And the thing is, getting rid of nitrogen is a much more involved project than getting rid of water or carbon dioxide. So now here's a figure from your textbook that shows some of the nitrogenous end products that animals generate. A number of these should be familiar already, although we haven't examined their molecular structure. So here's TMAO, for example, trimethylamine oxide, and up here we've got urea, and over here we've got ammonia, and down here we've got creatine as some examples. The thing is, each of these compounds has advantages and disadvantages for animals that produce them. And we've seen some of this already, as I mentioned, for the elasmobranch fish, which have to balance out high fluid urea levels, right, with high TMAO levels. And as some examples of some of these advantages and disadvantages, some of these are energetically cheaper to make, while others are lower in toxicity. And none of these is ideal all around, so different compounds are going to show up in different groups of animals. Also, as a reminder, nitrogen excretion isn't always accomplished just by the kidneys. So while mammals, birds, and the non-avian reptiles do excrete nitrogen via the kidneys, a lot of aquatic animals can excrete it across the gills or skin. So how to think our way through this diversity of strategies? Let's start out with what's considered to be the basal strategy, which is ammonia production. So animals that primarily excrete ammonia are called ammonotilic. Ammonia is the metabolically cheapest way to get rid of nitrogenous waste. Its production basically has no ATP cost. Also, as a side note, physiologists generally think of ammonia, so NH3, as equivalent to ammonium, since the interconversion just depends on pH. The thing is, although it's energetically cheap to produce, ammonia is extremely toxic. So that's actually what makes ammonia a really great household cleaning agent. When ammonia is inside an animal, even low concentrations can disrupt neuron function and tissue integrity. So animals that produce it have to be able to get rid of it quickly. And as a result, ammonotilism primarily shows up in aquatic animals, whether marine or freshwater. Since these animals can quickly release it into the environment and avoid any of these toxic buildups. In contrast, ammonotilism is rare on land, because it requires the excretion of a lot of water. There are just a couple of strange exceptions in terrestrial isopod crustaceans and terrestrial snails. And these guys actually void ammonia as a gas and then use that process to deposit calcium carbonate in their exoskeletons or shells. But those are the rare exceptions. So the next major type of strategy is called ureotelism for the production of urea, as we've discussed to some extent already. This strategy appears to have evolved with a transition to terrestrial life, so it shows up in most terrestrial vertebrates. So that includes mammals, some turtles, and some terrestrial invertebrates. And interestingly, amphibians switch from being ammoniotelic as juveniles and then become ureatelic as adults. Urea production is more costly than ammonia production, requiring between 4 to 5 ATP per urea molecule synthesized. But it is much less toxic. Such that steady-state blood concentrations of urea in ureatelists can be at least 20 times higher than steady-state concentrations of ammonia in ammoniotelists. So finally, the last nitrogenous waste excretion strategy we're going to consider is uricotelism for animals that primarily excrete uric acid. As shown here. So note that uric acid can occur in one of several different forms, but we're just going to show one form here. So uric acid is really interesting in that it and its derivatives are poorly soluble in water. Now, that might sound like a problem, but actually it can be an advantage because it means that if animals can get uric acid to precipitate out in the right location, they can excrete it as a paste or in a dry form instead of having to use any water to get rid of it. And further, a range of cations can also be incorporated into uric acid waste, such as sodium, potassium, ammonium ions, calcium, and magnesium. In addition, other purines or purine-derived compounds, so that includes the guanine, allantoin, and the allantoic acid that are shown here, these also behave similarly to uric acid, okay? And so uricotelism, or the production of other purines as principal waste products, is the most common strategy found in terrestrial animals. And this is really striking because it's even more energetically expensive to produce uric acid than to produce urea. But, that said, birds, lizards, and snakes all do this, along with other terrestrial invertebrates. And there are even some insects, snails, and land crabs that temporarily or permanently store purine wastes in their bodies. They don't actually excrete them because they don't have to. So this prevalence of purine waste production should actually make us ask a kind of different question. If this seems to work so well, then why don't the mammals do it? Why are the mammals over here? And the answer, as best we can figure, is that the urea concentrating capabilities of the mammalian kidneys sent mammals down one constrained evolutionary path, while the remaining animal groups simply took the alternate route. And this relies, generally biochemically, on relatively small modifications to ancient and universal pathways for things like the synthesis of the purine constituents of DNA. Okay, and there you have it. And I have to say that I hope at this point that you will never quite look at animal waste products in the same way ever again.